Lavanya ma'am. Good morning, dear students. Whether my voice is audible?
Yes, ma'am.
So today we are going to discuss on another most important concept, that is biotechnology and its applications. Right. So biotechnology is a branch of biology. We know that, where we are going to apply the technology for a research purpose. Fine. Deepti, I'm already answered. Thank you for 30-second question. Great. Now let us start. Please be interactive. Please message your answers, as well as you can tell your answer through mic also. Fine. Fine. So before going to that, before going directly into the questions, let me tell you which are the important concepts in that. GMO, that is genetically modified organisms like plants and animals, we have to focus on. Recombinant DNA technology we have to focus on. From that also, the question will be asked related to this concept. Okay, let us begin with the first question here. What does the second critical research area of biotechnology provide? Option A, optimum conditions for the catalyst. Option B, the pure form of a catalyst. I'm sorry. Option C, an impure form of a catalyst. Option D, purification of products.
Option A, optimum condition for the catalyst.
Ma'am.
Okay, Kanchana Raj told us option A. Samar, Veeresh, Soumya Naral. Okay, what about others? Dyuti and Suha. Fine. I think Dyuti, you were absent for 2 or 1 class. 2 classes or 1 class, I don't remember. Logit, option A. Fine. What about others? Be fast. You can answer through chat also. Just click on the screen of your mobile. You will get more option, otherwise directly chat option. In that you can, when you click, you will get the message box. In that you can type your answer. Kavita T. Rakshita. Very good. Okay. Parvati also. Okay. Most of them answered for option A. Saujanya. Okay. Fine. Anvita. Great. What does the second critical? E question artha enu antandre. We know that biotechnology is widely used in a research field. Right. So what does the second critical research area of biotechnology provide? Second critical research area. Let me check the answer. Option A is a right answer. So basically we have 4 critical areas related to research in biotechnology. The first one is providing the best catalyst in the form of improved organism. Usually A microbe or a pure enzyme. The second one is multiple ovulation transfer technology, MOET. Third one is create optimal condition, which you selected option A. Fourth one is downstream processing technology. So these are the four basic critical research areas in biotechnology. Good. Most of you, means all of them answered as option A. Let me tell you, while you are answering in the chat box, please don't copy the answers from the previous messages. Means you are cheating yourself if you copy it. That's it. The next question goes like this. What is the green revolution related to? Option A, milk. Option B, agriculture. Option C, water. Option D, animals. The PM answered through my
Option B, agriculture.
Good. Most of the answers are through mic and I got 9 chat answers. Rakshita, Deepti, Saumya Parvati, Veeresh, G. Okay. I don't know who is G. Dyuti. Fine. T. Rakshita, Preeti, Chaitra, Padmayan. Fine. Very easy question and quick responses. Great. The right answer is option B, that is agriculture. Okay. Let us read out the slide. Deepti, can you read out the slide, please?
Green revolution is associated with agriculture production. It is the period when agriculture of the country was converted into an industrial system due to the adoption of modern methods and techniques like the use of high-yielding variety seeds, tractors, irrigation facilities, pesticides, and fertilizers.
Okay, it is the period where agriculture was converted into an industrial system, Green Revolution. So we have few more questions on Green Revolution. Let us solve it. Who was the leader of Green Revolution? Verghese Kurien, Neil Kranti, Napoleon Bonaparte, Norman Borlaug.
Option D, Norman Borlaug.
Option D, Norman Borlaug. Okay. Only few chat answer, only 1, 2, 3, 5. Okay. Very easy question. 8 chat answers. Jyoti, Saujanya, Deepti, Chaitra, Anvita, Veere, Saumya, Naral. Okay, fine. Parvati. Okay. Right. Option D, Norman Borlaug is a right answer. He was the leader of Green Revolution when we consider a worldwide. Okay. Next. Fourth option. Who led the Green Revolution in India? Again, easy question. Option A, Neil Kranti.
MS Swaminathan.
Okay, Chaitra answered.
Option C, ma'am.
Fine, fine, fine. Great. Many answers I'm getting from the mic. I appreciate it. I got chat answers also from Parvati, Kavita, Saujanya, Dyuti, Veere, Saumya, Deepti, Yam. Then, yes. Fine. So option C, that is MS Swaminathan, is the right answer. Let us proceed to the next question, guys. Fifth question. Before that, let us read out some of the information regarding MS Swaminathan.
ಕೇಳಿಸ್ತಿಲ್ಲ.
Okay, fine. Let us continue. Just a minute. Fine. So fourth question we already discussed. Then fifth question. Okay, GMO stands for, very easy question. Option A, genetically matured organisms. Option B, genetically mental organs. Option C, genetically modified organisms. Option D, genetically mutant organism.
Option C, ma'am.
Genetically modified organism.
Or genetically
Option C, ma'am.
modified organism.
Option C,
Genetically
ma'am.
modified. Okay, great. Okay, fine. Option C, genetically modified organism is a right answer. It is not. Okay, it is not A, it is option C. Okay, sixth question goes like this. An important objective of biotechnology in area of agriculture is, please don't message or speak through a mic until I complete all the options. Until I read out all the options, please don't answer the question. Okay, an important objective of biotechnology. Biotechnology ಉದ್ದೇಶ ಏನು? ಯಾವ ಫೀಲ್ಡ್ ಅಲ್ಲಿ? ಅಗ್ರಿಕಲ್ಚರ್ ಅಲ್ಲಿ ಅದರ ಉದ್ದೇಶ ಏನು ಅಂತ ಕೇಳಿದ್ದಾರೆ. Option A, to decrease seed number. Option B, to produce pest-resistant varieties of plants. Option C, to increase phosphorus nitrogen production. Option D, to reduce the number of plants.
Option B, ma'am.
Option B, to produce pest resistance variety of plant, ma'am.
Fine. Padmayan, Kanchanaraj, Chaitra, Dyuti, Gayatri, Saumyanara, Samarth, Parvati, Veeresh, Akash. Fine, great. Very good. Option B, that is to produce pest-resistant varieties of plants, is the right answer. Fine. A GM crop is mature crop, infected crop, raised to green manure. D, transgenic crop. GM crop andre adar innond artha enu anta kelidare. What is the other name or other meaning of GM crop? Mature crop, infected crop, raised to green manure. D, transgenic crop.
Option D, ma'am.
Option D, ma'am.
Option D, that is transgenic crop. Veeresh, Parvati, Rakshita Joshi, Gayatri, Samarth, Deepti, Young. Then Padma, Chaitra. Okay, fine. So option D, transgenic crop, is the right answer. Next, option D. Fine. An example of abiotic stress is option A, pest. Option B, insect. Option C, drought. Option D, vectors. Abiotic
Option
means
C.
non-living.
Option C,
Option
ma'am.
C, ma'am.
Option C, drought. Okay. Samarth, Veeresh, Chaitra, Padmayan, Lakshmi, Saumyanara, Dyuti. Option C, drought, is the right answer. Other options are biotic stresses. Okay. Veeresh also answered as option C. Okay. Ninth question. GM rice is enriched with option A, option B, B, option C, A, option D, E. GM rice is enriched. Yes.
Option C, ma'am.
Option C, ma'am.
Option
Yes.
C, Soumya and Kanchanaraj answered through mic. What about chat answers? Deepti, Lohit, Lakshmi, Samarth, Dyuti, Suha, Parvati. Okay. Most of them answered as option C. Some of them have a confusion that they are voting for option A. Veeresh. ಆಪ್ಷನ್ ಸಿ GM ರೈಸ್ ಅಲ್ಲಿ ಯಾವ ವಿಟಮಿನ್ ಜಾಸ್ತಿ ಇರುತ್ತೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಹೌದಾ? ಸೊ ಲೆಟ್ us see let us check the answer. Most of them exam ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ ಗೊತ್ತಾ, even though they are knowing the answer, that is it is option C A vitamin ಅಂತ, ಆ A vitamin ಅದನ್ನ ಸೆಲೆಕ್ಟ್ ಮಾಡೋ ಬದಲು they will select option A in the confusion. Option A ಏನಿದೆ ಇಲ್ಲಿ K, that is vitamin K ಇದೆ. So option C vitamin A ಇದೆ. So option C is the right answer, but in the confusion they will right mark option A ತಲೆಯಲ್ಲಿ ಮಾಡ್ಕೊಂಡು option A ನ ಹಾಕಿದ್ರೆ ತಪ್ಪಾಗತ್ತೆ ಉತ್ತರ. So option C is the right answer, that is vitamin A. 10th question goes like this, guys. Bt toxin is produced by a bacterium called as Bacillus thuringiensis, Bacillus anthracis, Bacillus thermophilus, Bacillus subtilis.
Option A. Bacillus thuringiensis. Option A, ma'am.
Option A, ma'am.
Suha, Saumya Narar, Lohit, Samarth, Rakshita Joshi, Yuti, DPM, Viresh, Chaitra. So most of them answered as option A. Some of them answered as option C. Let us check the answer. Option A is the right answer. Padmayan, can you read out the slide, please? Padmayan, can you switch on your mic and read out the slide, please? Read out the slide. Kanchana Raj.
Yes, ma'am. Bacillus
Can you read out the
Bacillus thuringiensis (BT) is a gram-positive spore-forming bacterium that synthesizes parasporal crystalline inclusion containing cry and CYT protein, some of which are toxic against the wide range of insect orders, nematodes, and human cancer cells.
Okay, so in this we have to concentrate on the terms. Bacillus thuringiensis (BT) is a gram-positive bacteria. It is a bacteria which is gram-positive. Then it synthesizes parasporal crystalline inclusions which contains cry and CYT proteins. Cry and CRY and CYT proteins. Okay, that we have to remember. Next, GMOs can minimize the use of fertilizers. Genetically modified organisms, or here it is plants, can minimize the use of fertilizer. Whether this statement is true or false. Genetically modified plants ನಾವು cultivate ಮಾಡಿದ್ರೆ use of fertilizers ಕಡಿಮೆ ಆಗತ್ತೆ ಅಂತ ಹೇಳಿದ್ದಾರೆ. Whether this statement is true or false. ಸರಿಯೋ ತಪ್ಪೋ ಅಂತ ಕೇಳಿದ್ದಾರೆ. I got 10 chat answers.
Option A, true, ma'am.
Viresh, okay, Kanchana Raj, Saumya Narar, Viresh, Chaitra, Gayatri, Lakshmi, Padmayan, Suha TS. Fine. Most of them answered it correctly. That is option A. It is a true statement. So farmers can use less spray pesticides or fertilizers when they plant GMOs. This saves farmers money and reduces the amount of pesticides in the crop. Fine. Dash was the first transgenic crop. Option A, potato. Option B, brinjal. Option C, cotton. Option D, tobacco.
Option D.
Okay. Yes, option
Option D, ma'am.
Great. Samarth, Jyoti, Lohit, Parvati, then Lakshmi, Rakshita, Viresh, Padmayan. Fine. Option D, that is tobacco, is a right answer. The tobacco plant which produced in 1982. Just a minute. Yeah. The tobacco plant which was produced in 1982 was antibiotic resistant and was the first genetically modified crop. So in 1992, China became the first country to commercialize this tobacco plant. The first transgenic crop developed in India was BT brinjal. BT brinjal. Some strains of Bacillus thuringiensis can kill certain insects such as lepidopterans, scorpions, fruit fly, and dragonfly. Bacillus thuringiensis ಯಾವ insects ಅನ್ನ kill ಮಾಡೋ ಕೆಪಾಸಿಟಿಯನ್ನ ಹೊಂದಿರುತ್ತೆ? Lepidopterans. Okay. Yuti, Lakshmi Vadiyar, Lohit, Rakshita, Chaitra, Padmayan. Okay. Viresh, Rakshita. Option A, Lepidopterans. Lepidopterans is the right answer. Bacillus thuringiensis produce a protein crystals during a particular phase of their life cycle or their growth, which kill certain insects such as lepidopterans, adralli tobacco, budworm, armyworm anta baratte, coleopterans and dipterans, flies and mosquitoes. Fine. BT cotton is a cloned plant, transgenic plant, hybrid plant, mutated plant. BT cotton is a cloned plant, transgenic plant, hybrid plant, mutated plant.
Option B, ma'am.
Option B, transgenic plant. Samarth asked me a good question. He asked the common name of Bacillus thuringiensis. So here we don't have specific name for or a common name for bacteria. We will use them or we will use the name of the scientific name only to name them. Okay. Common name means we can say rod shape, spiral shape, like that. Okay, we can't say a common name. However, we have like for vegetables and all. For onion, Pisum sativum, for mango, Angifera indica. So we can't name, we don't have a common name like that for the bacteria. If you know the common name, please let me know so that I will also get a good knowledge of it. Fine. Most of them answered as option B, Viresh, Rakshita, Padmayan, Lakshmi, Yuti. Okay. Then, fine. Option B, transgenic plant is the right answer. 15th question. Insect-resistant transgenic cotton has been developed by inserting a piece of DNA from. I repeat, insect-resistant transgenic cotton has been developed by inserting a piece of DNA from an insect, wild relative of cotton, a virus, soil bacterium. BT cotton ಅನ್ನೋದು transgenic plant. ಅದರಲ್ಲಿ ನಾವೇನ್ ಮಾಡಿದ್ವಿ, ಇನ್ಸೆಕ್ಟ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ನ ಇನ್ಸರ್ಟ್ ಮಾಡಿದ್ವಿ. ಆ ಇನ್ಸೆಕ್ಟ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ನ ಯಾವ ಆರ್ಗ್ಯಾನಿಸಮ್ DNA ಇಂದ ನಾವು ಐಸೋಲೇಟ್ ಮಾಡಿದೇವೆ ಅನ್ನೋದನ್ನ ಹೇಳ್ಬೇಕು. ಆಪ್ಷನ್ ಎ ಇನ್ಸೆಕ್ಟ್, ಆಪ್ಷನ್ ಬಿ ವೈಲ್ಡ್ ರಿಲೇಟಿವ್ ಆಫ್ ಕಾಟನ್, option C virus, option D soil bacterium. I got 8 messages in the chat box. Okay, most of them.
Option D, ma'am.
Option D.
Option D, Padmayan. Option D, Parvati, Lohit, Dipti, Saundarya. Okay, Saundarya, Dipti. Fine, Viresh. Okay, so it is a soil bacterium. 16th question. The gene which was used to produce insect-resistant cotton plant was taken from, insect-resistant cotton plant. Now they are asking which organism. A. Anabena azole, B. Agrobacterium tumefaciens, C. Bacillus thuringiensis, D. Bacillus subtilis.
Option C, ma'am.
Option C.
Option C, ma'am. Option C, ma'am.
Option C. Okay. So option C, that is Bacillus thuringiensis, is the right answer. The 17th question goes like this. The protein crystals of B. thuringiensis contain toxic dash protein, bactericidal, fungicidal, insecticidal, antibiotic.
Option C, ma'am. Option C, ma'am.
Option C, ma'am.
Chaitra, option C. Lakshmi, Saundarya, Parvati, Padmayan, Viresh, Suhati, yes. Okay. That is option C. Yuti also answered as option C. Option C, insecticidal is the right answer. Let us read out the slide. Just a minute.
Soil bacterium Bacillus thuringiensis BT produces proteins that kill certain insects like Lepidopterans, tobacco dewworm, armyworm, coleopterans, beetles, and diapterans like flies and mosquitoes. Bacillus thuringiensis forms some intracellular protein crystals. These crystals contain a toxic insecticidal protein. The Bt toxin proteins exist as an inactive toxin, and it is converted into an active form due to the alkaline pH of the alimentary canal that substitutes the crystal. The activated toxin binds to the surface of midgut epithelial cells and create pores which causes cell swelling and lysis and finally cause death of the insect.
Thank you, Kanchana Raj, for reading voluntarily. So here we can get 2 or 3 questions. Please, I'm sharing the screen. Please go through the slide once again. All of you read this slide. And after that, related to this slide only, we have 2 questions in upcoming, slides. Okay, please read out this in your mind. Yes. Read out this. Take 2 minutes of time. Okay, let us proceed to the next question. The Bt toxin does not kill the bacillus because the Bt toxin protein exists as. They are asking Bt toxin enide adu bacillus anna bacteria anna kill madalla yake, because it contains what? Option A, lipid, option B, protoxins, option C, protein, option D, carbohydrate. Okay, someone asked me a doubt in between. Who's that?
From option B, protoxin.
Okay, fine. Yeah.
Option
Someone
B.
asked me a question. Okay, option B. Great. Someone asked me a question whether a butterfly will come in Lepidopteran. Yes, butterfly will also come in Lepidopterans. Fine. The Bt toxin does not kill the bacillus because the Bt toxin protein exists as inactive. Let me check the answer. Most of them voted for option B, that is protoxins. Great. Option B, protoxin is the right answer. Fine. The slide is same. To which cells does the activated Bt toxin bind? To which cells? Option A, neuroglial cells. Option B, lymphocytes. Option C, skeletal muscle cells. Option D, epithelial cells.
Option D, epithelial cells.
Okay. Sama, Dyuti, Saundarya, T. Rakshita, Saumyanara, Kanchana Raj, and few of them answered through mic. Deepti M, Rakshita, then Suhati, yes. Okay, Gayatri, Chaitra, and Parvati. So option D, that is midgut epithelial cells. It is activated or it binds to midgut of epithelial cells of the midgut. Next, 20th question. A gall-producing gene in Agrobacterium tumefaciens is a gall-producing gene. Option A, cry gene. Option B, Nef gene. Option C, tDNA. Option D, beta gene. Parvati,
Option
T.
A,
Rakshita,
Cryogena.
Option
okay,
A.
Kanchana, Raj,
Option A.
and Soundarya answered through mic. T. Rakshita, Chaitra, Samar, Padmayan, Saumyara, Yuti. What about others? Twentieth question, option A is the right answer. Cry gene. NIF gene, tDNA, beta gene are not related to this Agrobacterium tumefaciens. Option A is the right answer. Let's see. One nimsha. Agrobacterium tumefaciens. Then here this is Bacillus. Okay, fine. Okay. Just a minute. Soumya, Naral, can you read out the slide, please?
Yes, ma'am. Agrobacterium tumefaciens is a pathogenic bacterium that causes crown gall diseases, a plant tumor affecting a wide range of plant species. Crown galls develop upon transfer of a portion of the tumor-inducing plasmid. The transfer DNA, tRNA into the genome of bacterium's plant host.
Plant hosts. Okay, 1 minute. Option A is the right answer. The vector plasmid, when they ask, the answer will be tDNA, option C. Don't get confused. Next, T plasmid is found in Nitrosomonas, Escherichia, option C, Agrobacterium, option D, Bacillus. T
Option
plasmid.
C, Agrobacterium.
Option C, ma'am, agrobacterium.
Saumyanara, Deepti, M.
Option C.
Rakshita, Padma, Kanchana Raj, Dyuti. Option C, that is Agrobacterium, Samar. Okay, option C, Agrobacterium is the right answer. Fine. Veeresh also answered as option C. Toxic content of Bacillus thuringiensis is dash in nature, protein in nature, steroid in nature, amino acid in nature, alkaloid in nature.
Option D, ma'am, alkaloid in nature.
Option D, ma'am, protein.
Fine. Think over it. Toxic content, poison ಏನಿದೆ ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಅದು ಯಾವ ನೇಚರ್ ಅಲ್ಲಿ ಇರುತ್ತೆ? ಪ್ರೋಟೀನ್ ನೇಚರ್ ಅಲ್ಲಿ ಇದೆನಾ, ಸ್ಟೀರಾಯ್ಡ್ ಇದೆನಾ, ಅಮೈನೋ ಆಸಿಡ್ ಇದೆನಾ, ಅಲ್ಕಲಾಯ್ಡ್ ಇದೆನಾ?
Option D, ma'am.
Option D. Most of them are voting for option D. Some of them have a confusion with option A.
Option A, protein.
Okay. Just a minute. Option A, protein in nature is the right answer. Next, BT gene, which produces a protein toxic to insect larvae. Understand the question properly. It is a BT gene which produces protein poisonous substance. Insect larvae is tri, pri, arai, tri.
Cry.
Option D, ma'am.
Very easy question. Most of them answered through mic. Padmayan, Ditiyam, Saumyanara, Chaitra, Soundarya, and Nithi, Samarth, and Dyuti, T. Rakshita. Okay, option D, cry is a right answer. So Bacillus thuringiensis is a gram-positive spore-forming bacterium which synthesizes a crystalline inclusion which contains cry and CIT proteins, some of which toxic against a wide range of insect orders like nematodes, human cancer cells, etc. Option D is a right answer. Next, which nematode infects the roots of tobacco plant? Which nematode infect the roots of tobacco plant? Tobacco plant in a root anna yava nematode infect ಮಾಡುತ್ತೆ? Option A, Meloidogni incognitia, Didellenchus tipsaki, Zephenema index, Nacobus aberrans.
Option A, ma'am.
Option A, Lakshmi, Gayatri, Dyuti. Veeresh, Chaitra, Rakshita, Samarth. Okay. Padmayan. Okay, fine. Let me check the answer. Option A, that is Melodogene incognita, is the right answer. Okay. Fine. Deepti M, can you read out the slide? Deepti M. Slide ಓದಿ ಅಂತ ಅಂದ್ರೆ ಸ್ಲೈಡ್ ಅನ್ನ ಲೇನ್ ಬರ್ದಿರೋದು ಅದನ್ನ ಓದೋದು ಅಷ್ಟೇ. ನಿಮ್ಗೆ ಎಕ್ಸ್ಟ್ರಾ ಹೇಳಿ ಅಂತ ಹೇಳಿಲ್ಲ ನಾನು. ಎಸ್, anyone voluntarily who want to read the slide?
Yes, ma'am.
Yeah, sure.
Melodogene incognita, that is a root infecting nematode. It is also called as southern root knot nematode. The parasitic nematode has numerous plant hosts, including tobacco. It also usually attacks roots of tobacco and many others hosts plants.
Okay, thank you, Padma. So here it is a root infecting nematode. The other name of it is southern root knot nematode. Southern root knot nematode. So it usually affects the host plants like tobacco and the examples or the the plants related to that family. Option A is the right answer. I'm sorry. Which one is the method of cellular defense in animals and plants? Recombinant DNA technology, RNA interference, option C, immune response, option D, mutation.
Option B, ma'am, RNA interference.
RNA interference. Okay.
Option B,
Samarth, Rakshita, Padmayan, Saundarya, Veeresh. I'm sorry. Some of them answered as option D. Few of them voted for option A. Chaitra, Nidhi. Okay. Let us check the answer. Option B, that is RNA interference, is the right answer. Samarth, can you read out the slide? Just switch on your mic and read out the slide.
The method of cellular defense which work in all eukaryotic organisms is RNA interference. It is a biological process in which the RNA molecule inhibits the gene expression or translation by neutralizing the mRNA molecule. It is targeted for inhibiting the expression.
Okay. So here the method of cellular defense, we can observe it in all eukaryotic organism. It is a biological process. ಏನಾಗುತ್ತೆ ಅಲ್ಲಿ the RNA molecule inhibits the gene expression, inhibits the translation. ಹೇಗೆ? By neutralizing mRNA molecule. Neutralize ಮಾಡುತ್ತೆ mRNA molecule na, because translation should take place with the help of mRNA, right? It is targeted for inhibiting the expression. This is regarding RNA interference. RNAi stands for RNA intersection, RNA insertion, RNA intercom, RNA interference. Yes.
Option D, ma'am.
Option D, ma'am.
Option D.
Option D.
Soumyayan, Padmayan, Saundarya, Kanchana Raj voted for option D. What about the chat answers? Lakshmi, Deepti M, Parvati. Yes. Veeresh. What about others? Samarth? Fine. Let us check the answer. Option D, that is RNA interference, is the right answer. RNAi stands for RNA interference, which is the silencing of a gene expression. It is a silencing of a gene expression. Hege? By the administration of double-stranded RNA, dsRNA. Next. Dash is mobile genetic elements. Option A, viruses. Option B, bacteria. Option C, transposons. Option D, insertion sequences.
Option C.
Option A, ma'am.
Option C. Okay.
Option A, ma'am.
Option A. Fine. mobile genetic elements.
Option C, transposon.
Okay, transposons. Some of them answered as option C. Few of them voted for option A, that is virus. Then, okay, Rakshita, Samar, A. Jyoti, option C. Viresh, option C. Soundarya, not Viresh, someone. Okay. Yes, fine. Samarth, option C. Two of them again changed their answers, right? Okay. Let me check the answer. The right answer is option C, that is transposomes is the right answer. Transposomes means what? These are mobile genetic elements. These are segments of DNA. These are small segments of DNA which has a capacity to move around to different positions in the genome of a single cell. Okay, fine. Yeah. Rakshita and DPM also answered as option C. 28th question goes like this. Dash vectors are used for the introduction of nematode-specific genes in the host. Option A, bacterial. Option B, viral. Option C, agrobacterium. Option D, Melodognin incognitia.
Option C, agrobacterium.
Okay. ಯಾವ ವೆಕ್ಟರ್ಸ್ ಅನ್ನ ಇಂಟ್ರೊಡಕ್ಷನ್ ಆಫ್ ನಿಮ್ಯಾಟೋಡ್ ಸ್ಪೆಸಿಫಿಕ್ ಜೀನ್ಸ್ ಅನ್ನ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಕ್ಕೆ ಹೋಸ್ಟ್ ಅಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತಾರೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. So I got 11 messages. Lohit answered as option D. Samarth voted for option C. Most of them voted for option C, like P. Rakshita, Gayatri. Then, so 1 second, someone asked me a doubt here. Soundarya, Parvati, Saumyanara, Jyoti. Kanchanaraj asked me a question again in a message because your message got, I'm not finding your message here. Because I got many messages in between.
ಮ್ಯಾಮ್ ಯಾಕೆ ಅದನ್ನ ಮೊಬೈಲ್ ಜೆನೆಟಿಕ್ ಇದಂತ ಕರೀತಾರೆ? ಮೊಬೈಲ್ ಅನ್ನೋ ಟರ್ಮ್ ಏನಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ ಅಂತ ಮ್ಯಾಮ್ ಅಲ್ಲಿ?
Okay, ಇಲ್ಲಿ ನೋಡು ಒಂದು ನಿಮಿಷ. Transposomes are mobile genetic elements. Mobility means what? The meaning of mobility is movement. ಆ ಡೆಫಿನಿಷನ್ ನ ನೀನ್ ನೋಡಿದಾಗ ನಿಂಗೆ ಅರ್ಥ ಆಗತ್ತೆ. ಇದು ಸೆಗ್ಮೆಂಟ್ಸ್ ಆಫ್ DNA ಇದ್ರು ಇದರದ್ದು ಸ್ಪೆಸಿಫಿಕ್ ಅಥವಾ ಸ್ಪೆಷಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ಏನಂದ್ರೆ ಇಟ್ ಕ್ಯಾನ್ ಮೂವ್ ಅರೌಂಡ್ ಇನ್ ದ ಸೆಲ್ ಇನ್ ದ ಜೀನೋಮ್ ಆಫ್ ಅ ಸಿಂಗಲ್ ಸೆಲ್. ಅದು ಮೂವ್ಮೆಂಟ್ ಮಾಡೋ ಕೆಪಾಸಿಟಿಯನ್ನ ಹೊಂದಿದ್ರಿಂದ ಅದಕ್ಕೆ ಮೊಬಿಲಿಟಿ, ಮೊಬೈಲ್ ಜೆನೆಟಿಕ್ ಎಲಿಮೆಂಟ್ಸ್. ಮೊಬೈಲ್ ಮೀನ್ಸ್ ಇನ್ ಮೂವ್ಮೆಂಟ್ ವಿಚ್ ಕ್ಯಾನ್ ಮೂವ್ ಅಂತ. ಡಿಡ್ ಯು ಗೆಟ್?
Yes ma'am, thank you.
Yeah. Transposomes. Trans ಅನ್ನೋದು ಕೂಡ it also indicates somewhere movement. Fine, most of them answered as option C here, agrobacterium. Let me check the answer. Good question, Kanchana Raj. Keep it up. Option C, that is agrobacterium, is a right answer. Agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was successful. Why? Because it produced sense as well as antisense RNA in the host cell. Then, 29th question is, a method involving silencing of specific mRNA due to binding of complementary double-stranded RNA is called. Method involving silencing of specific mRNA. Silencing of specific mRNA silence ಮಾಡೋದ್ರಿಂದ due to the binding of complementary double-stranded RNA. ಆ process ಗೆ, ಆ method ಗೆ ಏನಂತ ಹೇಳ್ತಾರೆ? Option A, electrophoresis. Option B, RNAi. Option C, rDNA technology. Option D, DNAi. Yes?
Option B, ma'am, RNA interference.
Option B ma'am. Option B ma'am.
Saumyanara, Kanchana Raj, Padma N, or someone answered again. Soundarya, I guess. Okay, I got the message answers or chat answers as option B, option B. What about YouTube students? Chaitra, Deepti M, Saumyanara. Then, okay, most of them answered as option B. RNA interference is a right answer. So again, we can read out the definition here. It is a method of cellular defense in eukaryote. Cellular defense. It involves silencing of mRNA. Both of these RNA strands bind to each other and prevents translation of target gene. That is called as preventing translation of target gene is called as mRNA silencing. 30th question goes like this. Dash prevents translation of mRNA. Option A, enhancing. Option B, knockout. Option C, clearing. Option D, silencing.
Option D, ma'am.
Option D, ma'am. Silencing.
Option D. Parvati
Option D,
answered
ma'am.
as option D.
Option D, ma'am.
Okay. Okay, fine. Samarth, Veeresh, Saundarya, Parvati, Saumyanara, Jyoti, T. Rakshita, Padmayan. Fine. Most of them voted for option D. I'm sorry. Option D is the right answer. 31st question. In RNA interference, the DNA is introduced into a host plant in such a way that it produces both. Option A, complementary DNA and mRNA. Option B, mRNA and sense RNA. Option C, sense and antisense RNA. Option D,
Option C, ma'am.
DNA and RNA.
C, ma'am.
Option C. Okay, Nithi and someone told in the mic that option C. Anta. Stand answers I got
Option C,
as
ma'am.
option C again. Kanchana Raj answered as option C. What about others? Padmayan, option C. Okay. Veeresh, option C. Option C is the right answer. Okay. 32nd question. Sense and antisense RNA are dashed to each other. Similar, same, complementary, different. Where is the question?
Option C, ma'am. Option
Okay,
C, complement.
Saundarya. Okay, Chaitra, Samarth, Padmayan, Saundarya, Lakshmi Vadayar, T. Rakshita, Nithi.
Option C, ma'am.
Nithi.
Complementary.
Option C, complementary is the right answer. Yes, complementary is the right answer. Sense and antisense RNA forms. Sense and antisense RNA forms. Option A, dsRNA. Option B, ssRNA. Option C, dsRNA. Option D, ssRNA.
Option A,
Give me
ma'am.
the answer through the
Option A,
chat.
ma'am.
Just give me
Option
2
A,
minutes.
ma'am. Double-stranded RNA.
Okay. Most of them answered as option A. That is dsRNA, double-stranded RNA. It is the right answer. Next, 34th question. Formation of dash initiates RNAi process. Formation of dash initiates RNAi process.
Option D, ma'am. Double-stranded RNA.
Option A, dsRNA. Option B, ssRDNA. Option C, ssRNA. Option D, dsRNA. I got only 2 messages.
Option D.
Option D, ma'am.
Option
Double-stranded
yes.
RNA.
Option D, double-stranded RNA is the right answer, guys. 35th question. RNAi takes place in all prokaryotic organisms. RNA interference takes place in all prokaryotic organisms.
Option A, ma'am.
True or false?
Option A.
Option A. Most of them are saying it as option A, that is true. Okay. And some chat answers I got as option B, that is false. Right answer is option the A, that is true. RNA-like mechanism do exist in prokaryotes and seem to show functional analogies also. Then 36th question, which agriculturally important bacterial gene is isolated from rhizobium? Bacterial gene isolated from rhizobium. Okay, NIF gene, E gene, F gene, PDN.
Option C, ma'am.
Option C, F, Tina. Okay. Lohit answered as option A. Samath answered as option E gene. Okay, option B. What about chat answers? Soundarya, Veerish. The answer is option A, that is NIF gene. 37th question, delay in the ripening of tomato is due to option A, virulent gene, option B, antisense PG, option C, antibiotic resistance gene, option D, mutation. I repeat, delaying in ripening of tomato.
Option C,
Delaying in
antibiotic resistance gene.
okay, antibiotic resistance gene. Fine.
Option C, ma'am, antibiotic resistance gene.
Most of them are voted for option C. Chat answers also I got as option C. That is due to antibiotic resistance gene. Okay. 38th question, the transgenic tomato variety with an improved shelf life is, which is the transgenic tomato variety? Lyco 100, PHB tomato, BT tomato, flavor saver.
Option C, ma'am.
Option C, ma'am, BT tomato.
Option C, ma'am. Option C, ma'am.
Okay. What about chat answers? So here BT tomato, it can retain its texture and ferments up to 45 days. The 39th question, the transgenic plant flavor save tomato cries an artificial gene for. The transgenic plant flavor saver tomato carries which artificial gene? Delaying the ripening process, added flavors, both delaying in ripening and adding flavors. Option D, insect resistance.
Option C, ma'am.
Okay. Option C,
Option
Padma,
C,
Lohit.
ma'am.
Option C, Dyuti. Option C.
Option C, ma'am.
Both delaying and ripening process and adding flavors. Option C, ma'am.
Okay. Option C, both delaying and ripening of a process. Fine. Great. Option C is the right answer, both delaying and ripening process. 40th question, two bacteria found to be very useful in genetic engineering experiments are Nitrosomonas and Klebsiella, Nitrobacter, Azobacter, Escherichia and Agrobacterium, Rhizobium and Diplococcus.
Option C, ma'am.
Option C, ma'am. Escherichia and
Option C. Option C, Agrobacterium and Escherichia is the right answer. Fine. So this is regarding some questions on biotechnology and applications. Okay, most of you answered it correctly. Any doubt in the session?
No, ma'am.
Okay. Any suggestions for bio class regarding the question, regarding the explanation, regarding the method of interaction? Any suggestions? Do you want to say? If you want to say, you can say through message also. You can speak through mic also. Tomorrow's chapter, I will let you know through the message, Lohit. Okay, question Lohit suggested me. The question should be tough. Okay, fine. Yes, we will carry out the tough questions. Any other suggestion? Okay. Whether the way of interaction or way of explanation is comfortable for everyone. Tumba speed ಆಗಿ ಆಗ್ತಿದ್ರೆ ಹೇಳಿ. Okay then. We will adapt to some tough questions from the next class. Thank you for your suggestion. Have a nice day.
Thank you, ma'am.